So today I'll go briefly through some of our research and findings in the last 5 years or so, and how we're trying to understand colitis, most associated with swine dysentery, to fight antimicrobial resistance. And the idea is that we're able to develop preventative methods by understanding disease mechanisms. So when I talk about swine dysentery, I really enjoy this slide because, as Nathan said in my introduction, my last 10 years of my life have been dedicated to studying diarrhea. And some may laugh at that. I still find it interesting. But when you think about swine dysentery, this is probably what comes to your mind. Spirochetes here, gram-negative spirochetes, classically Brachyspira hyodysenteriae, more recently other species. If you walk into a pen, there is animals affected by swine dysentery or Brachyspira hyodysenteriae. This is your classic picture, is mucohemorrhagic diarrhea. There's a spectrum of diarrhea that comes with the disease, so you could eventually find anything from watery diarrhea to mucoid or mucohemorrhagic. Upon necropsy, this is the other classic picture you'll find, is a very angry looking colon. There is some, there's a spectrum, but most of the time you will see this photo over here, which is a hemorrhagic mucosa, some sloughing off of that superficial epithelial layer there. Usually macroscopically, all you see is sloughing off of small pieces of mucosa. But really, it all leads to diarrhea, and when any disease leads to diarrhea, it leads to poor animal performance, and that's why we're here today. We are concerned about animal performance, and we're concerned about how can we improve welfare so that we can fight this disease, make more money, and have happier, healthier pigs. In the past, when you thought about swine dysentery, if you're educated in the last 40 or 50 years, you probably thought about Brachyspira hyodysenteriae. That was our classic pathogen associated with swine dysentery, and it was almost pathognomonic. If you went into a barn, you saw mucohemorrhagic diarrhea, you would possibly think about Brachyspira right away. In the last 8 years or so, we got concerned about a novel Brachyspira that is not so novel anymore, and it's now called Brachyspira hampsonii. Brachyspira hampsonii causes a disease undistinguishable from a disease caused by Brachyspira hyodysenteriae. It's clinically the exact same thing. You can't tell them apart. If you see a pig inoculated with either of those two, it's the same thing. But we can tell apart the bugs. And we can say that although Brachyspira hyodysenteriae came first, just because that's where we reported it first, Brachyspira hampsonii has been around for quite a while. We just took a little bit longer to detect it. Other Brachyspira that are interesting for swine practitioners is Brachyspira peloscoli, leads to colonic spirocathosis, and it's a bit milder disease. Usually all you see will be some watery diarrhea, no blood, some mucus, not necessarily, but still cause reduced growth rates. So it affects production at the end of the day. Finally, two other important ones that are still somewhat debatable are Brachyspira swanetina and Brachyspira murdochii. There's some controversy when it comes to, are these two Brachyspira actually pathogenic to pigs? There are reports saying yes, they're very pathogenic, where B. murdochii and B. swanetina actually lead to mucohemorrhagic diarrhea. But at the same time, we have seen isolates of both these species that don't cause disease at all. So it's hard to understand. We're trying to elucidate that. Finally, Brachyspira intermedia and Brachyspira innocens don't really seem to be associated with disease in pigs at all. But please keep in mind that we call Brachyspira intermedia for a reason. It seems to be in between other things. So there are reports of Brachyspira intermedia causing disease as well, but not as bad as Brachyspira hyodysenteriae and hampsonii, that's for sure. Anyway, when we think about swine dysentery, this is usually what you would expect to see if a pig that was either inoculated experimentally or has a field case of it. Things will range from just loose stools in the colon to accumulation of mucus to a fibrinotic disease to our classic picture of mucohemorrhagic colitis. It's important to keep in mind that we call it swine dysentery, we call it bloody diarrhea, but it's a spectrum of a disease. Pigs may or may not progress through all of these steps, and they may or may not eventually reach bloody diarrhea. But there is a disease going on, and it's important to keep that in mind. But what do we care about Brachyspira hyodysenteriae and swine dysentery in North America, especially in the U.S.? We care about it a bit more in Canada because we have a different industry. But things are moving forward when it comes to Brachyspira hyacintheriae. There are multiple reports about Brachyspira hyacintheriae being identified from healthy pigs, especially multiplier units. And what that is and what that means is actually pose a bottleneck for animal trade. You can't be shipping B-hyo-positive pigs around the world. And that's not just in Australia, where this came from the David Hampson's group. We saw the exact same thing happen in the Netherlands. We saw the exact same thing happen in Canada. So apparently, Brachyspira hyacintheriae may not be always pathogenic. In addition to that, Brachyspira hyacintheriae obviously, just like any other bacteria, develops resistance to specific antimicrobials if the pressure is too much. We're able to select resistant strains. And you may or may not say, well, you know what, we have carbodox, we don't care about that right now. And I agree with you. If I was practicing in North America, I'll probably think that way. But FDA is moving towards banning carbodox because of carcinogenic residues. The EU, European Union, has already banned it. Can be used over there for animal treatment. Canada has already banned it. We can't use it either. So my prediction is that FDA will eventually reach to the same level that these other countries have, and carbodox will be no longer available for veterinary use, especially with pigs. Why do we care? It's because it costs the industry a lot of money. So if you can't treat your pigs, if you don't have an efficient treatment, there are no vaccines, and we'll talk about that in a bit, you start losing lots and lots of money. It is estimated that we're talking about something like $11 to $17 per pig affected within the system. So that is a lot of money at the end of the day. Brachyspira hyacintheriae, like I said, there are no vaccines. The OIE back in 2015 actually identified the disease as one of the high priority targets for the development of vaccines. As you may or may not notice down here, there is a specific comment saying technical barriers to vaccine development. That means we can't make a vaccine for this. We have known Brachyspira, we actually have known swine dysentery for almost 100 years now. We have known the causative agent Brachyspira hyacintheriae for 50 years, and we still don't have a vaccine for that. And it is a big challenge. When it comes to the disease, it's not just what we have learned in the past 100 years, I guess, is that it's not just having the bug. There's a whole balance here between host, environment, and pathogen that may or may not trigger disease. And that may explain why sometimes we have healthy animals that are colonized but are not sick. And the challenge for us here as practitioners is really, how can we break this chain and how can we move on to either stop disease or prevent disease without using or being a bit more judicious when it comes to antimicrobial use. So let me briefly tell you how Brachyspira causes disease and what do we know. And this is going to be a very short story. This is a pig colon. So these are the colonic crypts. In green here we have mucus. Here's the lumen. So if the ingesta would be passing through, it would be going this way. If you're an unlucky pig that is colonized or infected by Brachyspira hyacintheriae, it will swim through the mucus and start colonizing the mucus layer in your colonic crypts. It loves mucus, and it will eventually start colonizing your crypts. And when it does that, and this is a classic picture you see if you ever talk to a pathologist, what does it look like when a pig has swine dysentery? It looks like there's a bunch of spirochetes in their crypts. They multiply in that area. They seem to really like that specific niche, and they definitely are able to swim through the mucus layer. So that's a pretty inefficient barrier from the host side. Now, we don't fully understand what happens here. Is it that all we need is Brachyspira just being around epithelial cells, or is there a factor? And note here that I'm not talking about a toxin, I'm talking about a factor because really we have no idea what it is. But it seems there could be a factor that is expressed by this pathogen, and it leads to the loss of water and electrolytes to the lumen. And this configures diarrhea. In association with that, there's also loss of red blood cells, so that configures our bloody diarrhea. The main question here is, how does all this work? And that's the holy grail right now for Brachyspira researchers, is trying to understand the disease mechanism. We don't. Back in the 80s, there was someone very smart who decided to expose pigs to gnotobiotic pigs to Brachyspira hyacintheriae alone. What they noticed is that without that regular microbiome, Brachyspira hydroxyntheria doesn't seem to make a big damage at the end of the day. It was a very mild disease when compared to field cases. So we went on, and what we do know, though, is using Flux studies that Brachyspira leads to a malabsorptive diarrhea, which is not really surprising because at the end of the day, if you have your mucosal cells sloughing off, nothing's going to be absorbed, right? So it's pretty expected at the end of the day. Anyway, so we moved on and we did a few inoculation trials, and just to briefly explain to you what we did is pigs would come in from a Brachyspira-free source, but they're normal commercial pigs. They would be led to acclimatized for a week, and then at day 0 here, they would be inoculated with loads of Brachyspira. I'm talking about 10 to the 8 bacteria per mil of inoculum, and they would get at least 10 mil. And then we'll let them go on for 14 days. Our control pigs would usually remain healthy. Nothing would develop. That's great. We do a good job in biosecurity. In our inoculator group, however, a portion of this group would actually remain healthy, and that would usually be a smaller portion of the group. A larger portion of the group actually would develop our classic mucohemorrhagic diarrhea signs. So just to show you what I'm talking about, these two guys here, they are pen mates. They're the same age. They were both inoculated the same day by the same people, the same way. This guy here did not develop disease. This guy here developed disease. And they were both exposed to the same amount of environmental contamination. So hard to explain why, but we did think about what if the microbiome is playing a role here. That's something that's inherent to each one of those pigs, meaning everyone has a different microbiome. So maybe the difference between these two pigs here really is inside their guts. And microbiome was the factor here that would change the balance on the scale here and would lead to disease. So we're lucky enough, we're conducting these experimental inoculations. We collect our samples pre-inoculation and post-inoculation. These were fecal samples. We use a technology called high-throughput DNA sequencing that everyone probably heard about by now. But what this do is just sequence a lot of DNA. In our case, we chose to sequence a specific part of the bacterial genome that later on let us tell who this DNA belongs to. So, and that's done through bioinformatics. So at the end of the day, what we had was a microbiota profile. And I know we talk a lot about microbiota, but I would rather say bacteriome profile, because really, we're only profiling here the bacteria that are in this sample. We're not looking at virus or fungi or anything else, no archaea either. So we have a simplistic view, but we do have a lot of data to look at. So interestingly, but not surprising, is the fact that following inoculation, following disease, we could tell pigs that were sick in red versus pigs that were not sick in blue. What you're looking at here is a bar graph that shows the different phyla that we detected in their relative proportion. So if you, for example, the main phyla dominating all these microbiomes are Firmicutes and Bacteroidetes. Firmicutes in green, Bacteroidetes in red. So great, we use some fancy statistical tool here. It's called the Similarity matrix. And we can easily tell between if a pig was sick or not. And again, not expected, right? Totally expected, not surprising at all. But the real question came when we looked at the pre-inoculation samples. So really, is the pre-inoculation microcolonic or fecal microbiome a trigger or a predisposing factor to mucohemorrhagic diarrhea? And from this data set, what we learned is that no, it doesn't matter what microbiome we tell. Using the tools we did at the time, we cannot predict if you're going to get sick or not using your microbiome. So we're pretty intrigued, though, by the fact that some pigs got sick, some pigs did not get sick, and we decided to think a little bit outside the box. We said, all this time, since Brachyspira hydroxyntheria and swine dysentery have been studied, a lot of people and a lot of effort was put into Brachyspira, a lot of effort into the pathogen, but maybe not a lot of effort into the host response to the pathogen. So we wanted to study that in a bit more detail than has been done before. And the way we could do that would be to know exactly when Brachyspira got in contact with the colon, what was happening. We can't do that using a regular animal model, because by the time we see this, so by the time we see mucohemorrhagic diarrhea, between inoculation and clinical signs, the minimal amount of time we have seen is 4 days. In 4 days, a lot can happen. We couldn't use that as our control. So what we did is we decided to bring in colon from healthy animals that we know that did not have brachyspira clinical disease before or within that herd. And we literally developed a methodology to keep colon alive in a laboratory dish, kind of like colon in a dish methodology. So we cultured colon, the whole organ, all its tissue. And it was pretty amazing that with this ex vivo model or explant model, after 12 hours of exposure to Brachyspira hampsonii, histopathologically, the lesions seem remarkably similar. So we have sloughing off the superficial epithelial layer. We have this catarrhal exudate here, which contain mucus and necrotic cells. We did not observe elongation of colonic creds because probably 12 hours is just not enough time, which makes a lot of sense. But if you look at the superficial layer here from a clinical case compared to our explant model, things seem to be looking pretty similar. So we're quite happy about that. So we started dissecting what was going on there. And one of the first questions we asked was, what happens regarding pro-inflammatory molecules? What is the inflammation process and who's the driver for that process? So here we have explants that were inoculated with Brachyspira hyacinthuriae. After 8 hours, what we noticed is that there was obviously a peak production, a difference in production between B-hyos or treatment group, LPS, which is based on positive control, against the control group, which was inoculated with PBS. There was an increased expression of mRNA related to IL-1 alpha cytokine, which is basically a precursor for IL-1 beta, which is kind of the effective molecule. So this is really an early signaling molecule. Same thing for IL-8, a bit more for LPS than Brachyspira hyacinthuriae. When it came to Brachyspira hampsonii, we had the opportunity to do a time series study, and we noticed that the same thing was observed regarding IL-1 alpha, and we also noticed that within 2 hours of exposure, there was a TNF alpha response. And for you, you're probably saying, well, what does that mean? Like, why do we care about IL-1 alpha and TNF alpha? Those are just pro-inflammatory cytokines, pro-inflammatory molecules that help drive the inflammation. They're important, but they do not explain all this. Just with those 2 cytokines, we probably cannot tell you, yeah, that's why we're seeing this very angry-looking colon. So we decided to go and take a look at this whole problem from a different perspective. And the way we did it is really looking at the way information flows through the mammalian cell. So when you think about it, it all starts with DNA and RNA and protein. This all means potential. So with DNA, you may have the potential to generate RNA, and if the RNA is translated to protein, you may have the possible function happening if certain things fall into place. But really, RNA and protein are actually resemblance of function, so DNA is not. But at the end of the day, what we wanted to look at was metabolites, because they really represent whatever happened did happen. That was the actual activity. So the cell waste at the end of the day tell us a better story than the potential things associated with DNA, RNA, protein. So the way we study this is the use of genomics, transcriptomics for RNA, proteomics for protein, and metabolomics for metabolites. So for those of you who are not particularly interested in this technique, metabolomics is nothing but the similar thing to a DNA sequencer. We're just looking at hundreds and hundreds of metabolites from a specific source, for example, in this case, from colon tissue, and we're able to profile them, and we're able to figure out amounts as well. So we can compare metabolite profiles between samples, which was pretty exciting. Problem is, what you end up at the end of the day is this massive amount of data that not only you're trying to figure out if your data actually shows you something significant, you're actually trying to figure out if it makes sense within all these metabolic pathways that we have identified and characterized since the early days of science. So it's a very laborious work, and it requires really a lot of bioinformatics, a lot of computer work, but also someone to sit there and interpret all that data. So that at the end of the day, you may end up with 1 or 2 things that actually make sense. So not really rewarding work at the beginning, but for us, we found these 2 amino acids here that were particularly interesting. So arginine seemed to be depleted and B-hyo exposed explants. So there was less arginine in the BHIO-exposed excellence than the PBS, or negative control, or LPS, or positive control. And then when it comes to L-citrulline, we saw the reverse. So BHIO-exposed explants had an accumulation of citrulline, while the other two groups did not observe that. So less arginine and more citrulline. How does that help us understand colitis? That's a very good question. So citrulline is associated with arginine biosynthesis. Doesn't really go towards that. We don't have a liver in our model, so the urea cycle here probably should be checked out of this model. But it does take part in the nitric oxide cycle. And that was quite interesting when we figured that out. And I'll tell you why. If you're a pig and this is your colonic crypt and you are infected by Brachyspira, Brachyspira will move on into your colonic crypt and will start exchanging or crosstalk with your cells. Like I said, we're not quite sure if it's the simple presence of the bacteria in the crypt or the actual expression of this factor X. And again, don't think about a toxin yet. We're not quite sure it's a toxin. Could be a microRNA. We don't know. Let's just call it factor X. But we know that once this happens, there is a pro-inflammatory response based on TNF-alpha and IL-1-alpha. And this pro-inflammatory response obviously recruits pro-inflammatory cells and leads to a pretty remarkable response in the colon. But importantly is the fact that it seems to activate nitric oxide synthase. This is an enzyme present throughout the colon, and it happens actually in a basal amount. So you have nitric oxide being produced right now on your gut lining. It doesn't mean much. It only means much when there is an overproduction of that. When we have an overproduction of this enzyme, it takes arginine and O2 to produce citrulline and nitric oxide. And surprise, surprise, nitric oxide, when overexpressed in the colonic epithelial cells, may lead to inflammation and necrosis, may lead to increased production of mucus, which help us explain part of the problem here, may also lead to vasodilation, which could help us explain why red blood cells are leaking towards the lumen, and finally, may lead to the opening of important chloride channels, such as CFTR. So if we have chloride channels opening at these epithelial cells in the gut, and chloride ions are leaking towards the lumen, this is driving the movement of water as well. So at the end of the day, this molecule could help us explain why we see mucohemorrhagic diarrhea followed by Brachyspira infection. Now, you as a practitioner, you're probably asking right now, okay, great. It seems like you draw some pretty nice diagrams. At least I would like to think so. But what does that matter to me at the end of the day as a practitioner, right? I don't really, so it's a nice picture. Well, if we're able to understand why this is happening, we may be able to develop other tools beyond antimicrobials. We may be able to develop compounds to target specific parts of that mechanism and hopefully be able to prevent disease. If we're not able to prevent disease, we may be able to treat disease without using antimicrobials, because we're not really going to be targeting the bacteria itself. We may be targeting something else. And at the end of the day, the goal is that we stop or reduce the amount of antimicrobials to treat the disease. Like I said, in North America, you guys are a bit spoiled still that you get to use Carbadox. If you ever have a chance to visit Europe, especially the Netherlands or Belgium, we do struggle a lot with diarrhea associated with Brachyspira. And that's not just Hyoscyaria or Hampsoniae. It goes to Pelosi coli as well. And when you lack some of these tools, you need to get creative. You need to look for alternatives. So if a vaccine may be a little too far, maybe we should be thinking outside the box and looking for other things we can use to prevent and treat disease. Just want to take the time here and obviously acknowledge that I did not do all this work alone. I had a lot of support. This went from Canada to Brazil, some people in Michigan there. Michael Dame actually was amazing helping us figure out some of this model. And also my crew at Utrecht University and the funding agency. Without them, this would never have happened.